I'm going to talk about something a little different. I'm going to speak about archaea. In particular, I'm going to speak about the Euryarchaea and how they arrange their chromosomes in the cell and the mechanisms that are responsible for this organization. And so,
je sais pas si moi, j'entends pas très fort en fait, mais je ne sais pas si c'est vrai.
Okay, so yeah, so first I just wanted to go give a little background. So in the late 1970s, the phylogenetic tree of life had a massive overhaul because up until that point, anything that was not eukaryotic was put into this prokaryotic domain. And then in the 70s, it was discovered that there was actually this archaea domain. And over the next 10 years, it became quite evident that actually archaea were much more similar to eukaryotes because they shared quite a lot of features in common. So for example, histones and also the RNA polymerase complexity. And so this continued with the advent of 16S ribosomal RNA sequencing and metagenomics with the identification. And so at first, the archaea were organized into the Euryarchaea and the Crenarchaea. But then over time, and now up to date, we have discovered 3,500, well, more than 3,500 genomes of the archaea. And this has been much more complex and much more diverse. So now the tree is kind of organized into these three main superphyla. So you have the DPA-NN, the Asgard, and the TAC, with the TAC being including the Crenarchaea. And so again, it's quite difficult to study the chromosome organization of these or even the subcellular organization because they're all extremophiles. So they are used to growing in, say, high salt or high temperature or an acidic environment. So they're quite difficult to study in the lab. So there aren't very many models that we can look at. However, there are some. So there's mostly they're from the Crenarchaea or the Euryarchaea. So the models for both are Sulfolobus and Halotherax. And they're quite different. So for example, most Euryarchaea are polyploid and contain histones, whereas that's the opposite for the Crenarchaea. Also, there is no defined cell cycle in the Euryarchaea, but actually the Crenarchaea have a very defined G1 and G2 phase. And what will become important later is the fact that when it gets down to the organization of chromosomes, the Crenarchaea actually have no SMC protein or canonical SMC, whereas the Euryarchaea do, and that's shared throughout the archaea kingdom mostly, and also with bacteria and eukaryotes. And so understanding the chromosome organization was limited because of the lack of tools, but this changed last year when Steve Bell's lab published the first high-C map of an archaeal species, and it was the Crenarchaea sulfolobus, or they did 2 species. And they performed high-C and they obtained a 30 kb resolution. And one of the most important things they saw was that although there were domains, they were quite large and they were much more similar to eukaryotes. And they saw that there was this plate patterning in the high-C map, which they identified to be A and B compartments. They also found an SMC-like protein. As I said, they didn't have an SMC, but they found this coalescin or CLIS-N. And when they performed ChIP-seq along the genome, they found that CLIS-N typically bound to the B compartment, so the transcription inactive compartments. And it was actually responsible for this, so this is a Shrek representation of the high-C map. It was responsible for the coalescing these B compartments together and actually was in control of this organization of the cell. So it promotes the B compartments. They went one step further and showed that when they inhibited transcription with the actinomycin D, in the high-C map, you lost this plate pattern. You had a redistribution of the CLIS-N binding across the genome. So for example, it was lost in this B compartment. And as a result, you had a weakening of this compartmentalization signal. And so they concluded that transcription was really driving this structure, and changes in transcription mediated CLIS-N binding, and this caused a change in the chromosome organization. What was interesting to us when this paper came out, because it came out in Cell, so it was widely seen, and also it had a very general title where it said that basically, if you read it one way, that all archaea kind of have this compartmentalization. However, we know that even in bacteria and eukaryotes, you can't make those sorts of generalizations, and something like archaea, which is so much more diverse, we wondered whether actually this was true for all archaea, and do other structure and mechanisms exist, and also, for example, how are the Euryarchaea structured? At the same time as us asking this question, we had also developed a new improved high-C protocol, which for the first time in bacteria offered a sub-kilobase resolution. And so Here I will just show an example of what the technique does, but we have E. coli, so this is the genome map. But what becomes really interesting is when you go down, so this is a 50 kb window in a published E. coli dataset and our new method, and this is 500 base pair bins, and you can see now it really allows the opportunity to look at structures that correlate to genes and operons within the cell. And so when we had this method and these questions, we thought, why not put them together and try the bacterial protocol on the archaea? And so that's what we did. And so we teamed up with Roxanne Lestini, and who is an expert in Halotherix volcanii, which is a Euryarchaea. And it was quite interesting for us because obviously the Crenarchaea, the sulfolobus, is all the way over here in the TAC, whereas the Euryarchaea are quite diverse and in the over here in, sorry, the Halotherix or Euryarchaea, so they're quite far away in the phylogenetic tree. The genomes consist of 4 chromosomes, which I'll go through in a minute. But it's interesting to note that it's very GC rich, but it does contain these AT rich islands, and it's also a polyploid. So we did the first high C map, and this is what we got. So it was really cool. I mean, you can immediately see it's so much better than what the sulfolobus looked like. So we can actually go down to 1 kb resolution, which is a 30-fold increase compared to what we've seen in the cell paper. And just to talk you through the chromosome, we have the genome. We have this main chromosome in white, and then we have a PHV3 chromosome, a PHV1 chromosome. There was a PHV2 plasmid, but that's been lost, and that was published, so we knew that was the case. And also the PHV4 chromosome has integrated into the main chromosome. And again, that was known before we started. So just to talk you through some of the features we identified with this new like really high resolution high C, we identified domains throughout the chromosome. So at the larger scale, there's about 10 domains that are structured between 70 and 520 kb. And then we have, when we look at smaller structures, we have about 23 domains, and they're sized about the same as what you would see in bacteria. So they didn't see these sort of domains in the sulfolobus with their method. When we then performed RNA-seq on these cells, and when we correlated the high C contacts in blue with the transcription, we saw that there was a good correlation between the two. Again, not unexpected. We've seen that before in other organisms. But what was cool with our technique was when we went down to a 1 kb resolution in these cells, we could really see the correlation between transcription and smaller structures, so operons and genes. And so here you can see where you have regions of high transcription, and it correlates with high C signal. And so then the third observation we made was, and it was kind of hard to miss, were these played patterns that are throughout the genome. And so these are very similar, like they're also, we saw played patterns in eukaryotes and in the Crenarchaea, but these are much smaller. They're only 2 to 15 kb in size. And upon first inspection, they really seem to correlate with regions of, as I said, there's these AT rich islands. So if we look at the genome GC content down here, you can see that there are dips. Mostly it's about 70%, but sometimes it dips down to about 50 or 45. And you can see it much better when we look at the smaller chromosomes and the integrated PHV4 chromosome, and we zoom in. So you can see that where you have these played patterns, they correlate with regions that are more AT rich. And interestingly, there's a lot of insertion elements in the genome. So we can see that these AT rich regions are dense with these IS elements. And so here where we have about 50 of the total genome IS elements, there's 102 in total, we can see this pattern. And then as a control, if we look at the PHV3 chromosome, there's quite a few less. And you have a much less of that played patterning. And then in the PHV1, you have a lot for such a small space. It's only 85 kb. And for that reason, you have a really poor high C matrix with lots of gaps, etc. And so we do have, and the fourth thing we saw, which was actually really interesting, was we recognized DNA loops throughout the chromosome. And so we utilized a method that's recently been published in the lab called Chromosite. And I won't go into the details, but the reference is there, and it's a really cool paper. So it's a computer vision based approach, and it detects patterns in high C maps. And in the Halotherix maps, it detected both DNA loops and chromosomal borders. And so the border number correlated quite closely with our initial analysis, where we'd recognized these 23 borders. So there's 26 with Chromosite. But what was interesting was we detected 64 loops throughout the genome. And I'll come back to this later because we explored a relationship with them and between them and transcription. And so having this map, the first thing we wanted to see was whether there was any compartments. So because there was clay patterning in both the sulfolobus and the halothorax. And so in sulfolobus, as I said, they tested this by treating with the actinomycin D, and they'd shown that they'd been completely lost. So we performed the same experiment, and we also performed RNA-seq and the compartment analysis. And so in the actinomycin D cells, it's the same as any time you inhibit transcription, you lose short to mid-range contacts. So this is shown in the ratio map. But what we didn't see was relative to the rest of the signal, the compartment signal was still there. It was just weakened to the same extent. Whereas you can see the transcription is really altered. But interestingly, the compartment index isn't that different, and it seems to be the same. So what we concluded from that was that it does seem to be that there's these structures, but they're not the same. They're not transcriptionally active A and B compartments, it seems. You can see this a bit more when you look into Zoom. So for example, here we have exponential stationary and actinomycin D treated cells. Stationary also has a change in its transcription relative to exponential. And you can see that although you have changes in transcription and changes in structuring, you don't really change this compartment. And so this is the opposite to what was seen in the sulfolobus. One other thing that, as I said, we detected these loop-like structures. And so we noticed when we looked at our transcription cells treated with actinomycin D and also stationary phase, that you lost these DNA loops, and you also lost DNA borders. And so we implemented chromocyte again. So here we have the controls for both loops and borders. And you can see in both the actinomycin D and stationary, you have a loss in both borders and loops. So from this, we concluded that transcription doesn't drive the compartmentalization. Instead, it drives these border structures and the DNA looping in the cell. So because we'd seen these loops and the link with transcription, it's been shown before that SMC-dependent loops have been identified in eukaryotes and in some bacteria. So we asked the question, are these loops mediated by SMC and halothorax? The SMCs are really conserved in the archaea. So for example, there's free SMC or SMC-like proteins in halothorax volcanii. The SMC protein itself is conserved throughout the Euryarchaea, the Asgard, and some of the TAC. The RAD50 is just kind of confined mostly to the Euryarchaea, and SPH4 seems to be much strictly kept to the halobacteria. And so we constructed deletion mutants for all three. And to our surprise, we observed no significant growth defects in any of the mutants. So here are the generation times, and within error, they're approximately the same. And also, when we did microscopy, the cells have a normal morphology and also a normal DNA content. So there's no anucleated cells or anything. They seem completely happy. So we were quite surprised when we performed high C on them. And so this is the delta SMC versus the wild type. And this is the ratio map of the two. So as you can see that we really lose several borders in the SMC mutant, which can kind of be seen in the maps themselves. So if you look at this border here, in the SMC it's lost and it's almost shifted. And so we were really interested in this, and because of the link between SMC, the loops and the transcription, we decided to check the transcriptome of these cells. And again, to our surprise, we saw that actually when we compared differential expression analysis of SMC mutants over wild type, less than 5% of genes show a greater than twofold change. Like they don't seem to care. And actually only one of the borders, where I've shown here on the right, actually correlated with a significant loss of transcription. So it seems to be that we've looked at all of these borders and there's no consensus sequence, there's no relation to the genes that are at these borders that would cause these specific borders to be affected. And so we're still investigating this, but it's highly conserved between replicates. So I think it's going to be really interesting. Just to kind of show you the controls, we also did the RAD50 and the SPH4. And although there are some changes in the genome architecture of these mutants, they're quite subtle and we're still investigating them separately, but they don't seem to have the same phenotype as the delta SMC. And so just to kind of show that in better quality, so we have the wild type and the free mutants here. And you can see in the SMC at this border, it's really shifted and lost, whereas you don't see the same in the RAD50 and the SPH4. And that's kind of also kind of further highlighted with the ratios underneath for each comparison. What we do see with the SMC however, is like transcription, we have a loss of chromosome borders. And so again, we've used chromocyte to detect and quantify those. And we see that in both, in SMC mutant cells, we lose both the borders and the loops, but we do not see the same loss in either the RAD50 and the SBH4. And so it wasn't super surprising for RAD50 because it's previously been shown to work with mRA11 in DNA repair in these cells. And so it seems to be that maybe in Halotherics volcanii, like the only true SMC is SMC. So yeah, so we had these, just to conclude what we'd seen from the Halotherax. So we identified three main features: these self-interacting domains, the played patterning, which was not linked to AB, transcription-mediated compartmentalization, and these DNA loops. But as I'd said before, one of the things we did notice was this played patterning did seem to correlate with AT-rich IS-dense elements, and so we wanted to test that further, and rather than generalize with just one particular archaeal species, we looked at two more. So we did Halobacterium salinarum, which is another halophile, so it's very similar to Halotherax. It's also got multiple chromosomes, and it's also got a G-rich chromosome, which is genome, which has AT islands and a lot of IS elements. Conversely, Firmicockus kodakaraensis, which is quite far in the Euryarchaea tree, is actually a hyperthermophile, so similar to the sulfolobus species. It's also a strict anaerobe, so it's much more difficult to deal with, but it's also polyploid. But interestingly, it has a stable GC content, and it also doesn't have very many of these IS elements. And so we performed Hi-C, and so on the Halobacterium salinarum, we see that they have very many similar features to the Halotherax. We identify domains, we can see DNA loops, so I've shown that with the chromocyte overlay. But also, and what's important for us is it does have a played pattern, but not really in the main chromosome. It's only in the two smaller chromosomes, the PNRC 100 and 200. It's worth noting that these two blank spaces are because both of these mini chromosomes share a 145 kb sequence. So we can't map that accurately. And so, yeah, when you look at this played patterning and you look at the GC at the bottom, it really correlates, and it also correlates with the presence of these IS elements again. And so this kind of confirmed to us that maybe that was what was responsible for the played patterning in Halotherax. And furthermore, when we looked at Firmicockus kodakaraensis, although the map is less defined, because I think it's much harder to work with because it is this thermophile and a hyperthermophile, and it is an anaerobe, so it's harder to cross-link. But we could get a 5 kb map, and we did identify some structures. But what was interesting was we didn't see any played patterning, and this kind of goes again with the fact we have this stable GC content and only 7 of these IS elements throughout the entire genome. And so,
sorry.
And so just to conclude on this, we have this super cool Hi-C technique now that works in both bacteria and eukaryotes. And because it's Hi-C and because you just need to cross-link your cells, it's quite good at studying. It would be very useful for studying non-lab models and quite difficult models. And also precious samples, because we can now use a tenth of the material that previous Hi-C methods have used. But it's also, what it's shown is because we've been able to use it to look at 3 different Euryarchaeal genomes, and this is the sulfolobus, but we've reprocessed it using the same method, so we can compare between the two, that it's kind of dangerous to always make generalizations, especially when you've only looked at one model of an entire domain. And so we don't see transcription-dependent compartmentalization in the Euryarchaea, and obviously it's been seen in the Crenarchaea. But instead, we do see that transcription is driving the formation of these chromosome borders and loops. And so actually, they all have the same players. Like they have the SMC-like protein in the Crenarchaea, and we have SMC in these organisms, and we have transcription that's driving structures, but they're doing different jobs. And so that's again reflective of the entire bacteria and eukaryotes as well. We see the same players, but we see different structures because of it. And so it'll be interesting to keep studying with this technique, different organisms, because we expect the diversity of these structures and the mechanisms responsible to continue to increase. And so finally, just to acknowledge who did the work. So Roxanne Lastini was instrumental. She did all of the work with me for the Halotherics volcanii. Aurora Gorlas gave us the Firmicockus kodakaraensis. And I would like to thank Roman Kazul and the rest of the RSG lab, and I thank you for your attention, and I'll take questions.
Okay, thank you very much. So questions? I have a questions. So regarding the histone-like, the nucleosomes in your species, I think that you have I can't hear you. You have nucleosomes in the species where you have plate patterns, no?
You have them in the Euryarchaea, but you don't have them in the Crenarchaea. So that's what's interesting, because the Crenarchaea seem to have this compartmentalization, which is much more eukaryotic, but they don't have any histones. Okay. Whereas we see whereas it's the opposite. The Euryarchaea have the histones, but they don't seem to have the compartmentalization. So it's really intriguing to see that it's different between the two.
Could you imagine to have a map like a macro-C, to have both maps of nucleosomes and IC to?
Yeah, that's a really good point, because I have a feeling that perhaps the plate pattern we're seeing in the Euryarchaea and Halithorax might correspond with the fact that you have less histones binding in the AT-rich regions compared to the GC-rich.
Yeah, that's because there is a correlation between AT-rich sequence are less prone to.
Yeah, and actually you say that, but it's really funny. Last night I realized that someone's actually done that experiment. So it would be nice to realign their data with our maps and see whether there's a correlation.
Okay, thanks a lot. Geneviève?
Hi, very nice talk, thank you. I was having a question about the concentration of IS element at your AT-rich island. In bacteria, it was suggested that IRS actually bind GRAS,
yeah.
Right? Do you have some evidence that it is the case also here, or that an enzyme could also be involved in this kind of insulation?
Directly in the Halithorax, we don't, because little is known. But actually, if we go to, for example, go back to bacteria, like you said, and you look at something like Deinococcus radiodurans, it's also an extremophile and it's also a polyploid. And actually, they found, for example, when you delete SMC, so the cells don't care, the same as the Halithorax, except for when you basically treat with a gyrase inhibitor. So perhaps it is something similar along those lines, but at the moment, no data has been done for that in Halithorax, so it's hard to tell specifically. It's quite complicated because you have all these, like the SMC observation. It's interesting, but so much of the genome doesn't have a function yet. Like the genes are known, but they're uncharacterized, so it gets a bit frustrating at that point. But no, it's a good thing to look into.
It might be that you may find some inhibitors of GRAS that may be conserved and would work too, and it might,
Yeah, no, it's a very good point. That I'd have to check with Roxanne because she's the expert, but I know that most of the antibiotics that work on bacteria don't work on the
don't work. Yeah. Okay.
So, but we can look into it and for sure try it. So.
Yes. Thank you. Thank you so much.
Okay. Emmanuel?
Yes, hello, Charlie. These archaeas are really funny. I had a question. I had the impression that when you use actinomycin, you use most of the loops, but some are remaining.
Yeah.
So I was wondering what are they?
I think it might be because the treatment we're using is actually, in comparison to other people, is quite low. So when you look at the RNA-seq, there's still some genes that are still quite high, like maybe because they have stable mRNAs, I don't know. But I wonder whether it's because if we were to use either a longer time or maybe increased concentration, we'd actually get rid of all of them. I think it's maybe that some genes are still being expressed and it's linked to that. But no, it's a good point. We don't completely abolish the loops.
Okay, thank you.
Thanks.
Thank you.
Okay, Aurel?
Hi, Charlie.
Hey,
great talk. I was wondering, so the last archaea you showed, I can't remember the name, but it seems to be much more featureless. When you look at the diagonal, there is no loops or border, it seems. So why do you think that's the case? What's missing in this one?
To be honest, it's a very good question because it's something we've tried the cross-linking step in many ways. Because they grow at 85, we obviously can't add the formaldehyde in at 85 because it won't cross-link. So we tried adding it to cold formaldehyde, or, you know, spinning the cells down, which is what Steve Bell does, and resuspending in PBS formaldehyde. And every time the map looks exactly the same. So now we're like, well, is it because of the technique, or is it because of there's just no features? But yeah, until it's quite hard. So I think we're going to try and optimize the cross-linking step to see whether we can improve on things, because it seems hard to believe it would be that flat, to be honest.
Yeah, it doesn't like SMC, for example, or
no, it doesn't.
Yes. Okay.
No, but no. All the Euryarchaea, they mostly have the SMC. So I think, because I think it's going to have the same structures. I think it's just that we need to improve the technique a little bit on the cross-linking step.
Thanks.
Okay, so no more questions. So thank you very much, Charlie.
Thanks everyone for listening.
And on peut remercier tous les orateurs de ce matin.